Good morning. Today we are going to talk about Salivaomics, saliva exosomics and saliva liquid biopsy. In the last seminar we were talking about the salivary biomarkers and how those salivary biomarkers were useful in identifying systemic as well as oral diseases. Today we are going to see about Salivaomics, saliva exosomics and saliva liquid biopsy. Many diseases have been diagnosed via detection of biomarkers in the blood, that we know. But saliva diagnostics could offer a less invasive and more reproducible alternative. So routine saliva testing of at-risk patients may reduce cancer-associated mortality. So when there is a set of patients who are at risk, if we just do a non-invasive salivary test to check the risk assessment, we can select the pre-symptomatic saliva testing and the early treatment for the patients with head and neck cancer and thereby improve patient outcomes. So at-risk patients as well as the pre-symptomatic saliva testing can help in improving the patient outcomes. And the lack of basic mechanistic understanding of how the disease-related biomarkers reach the saliva still limits the translation of salivary diagnostics from bench to bedside. So we don't know how the salivary biomarkers reach the saliva from the disease-related areas. So a US-based consortium had generated a comprehensive catalogue of the salivary proteome of healthy people, revealing that 20% to 30% of the salivary proteome overlaps with the plasma proteome. So this US-based consortium has given a statement that 20 to 30% of the salivary proteome resembles the plasma proteome. So the close physical proximity of the blood suggests that many salivary constituents are derived from the blood. In the anatomy and physiology of the salivary secretion, when we saw that the acinar cells are lined by the blood capillaries through which the salivary secretion happens. So the close proximity of saliva and the blood is a big advantage for the biomarkers to reach the saliva. So the academic and industry sectors are working on three core elements of saliva-based diagnosis. And those are nothing but the Salivaomics, saliva exosomics and the saliva liquid biopsy. So first we will see about Salivaomics. So Salivaomics is defined as the integrative study of saliva and its constituents and functions using omics technology. So high throughput methodologies enable researchers to identify DNA, RNA, protein and metabolite based biomarkers via omics studies, and they also facilitate the study of salivary microbiomes. So what is high throughput methodologies? It is a technique which uses robotics or other high methodologies to screen large number of samples simultaneously. So they can screen large number of samples simultaneously for all these omics studies. So in that way we can get large number of data, compare those and then decide, make a data set. So other elements in the oral cavity can add to the biological notch the saliva sample. So we know that saliva is collected from the oral cavity which has food, oral bacteria and other things which can make some disturbance in the salivary biomarker detection. So the process of mastication can alter the composition of saliva as well. So this is the drawback in the Salivaomics when we take only saliva as the main component. So here there is a map, proof of concept that saliva biopsies can be exploited for biomarker analysis, and this shows that what all biofluids can be used and what all condition can be detected and what are the sources for the RNA. So in the biofluids we have sputum, which is the thing, and saliva is also an important part of these biofluids. So when we talk about saliva exosomics, saliva exosomics is an emerging subfield of Salivaomics research, and its focus is the analysis of the molecular cargoes carried by the small membrane enclosed extracellular structures called as exosomes. So they are 30 to 100 nanometers in diameter, and these exosomes are surrounded by a phospholipid bilayer carrying many cell type specific cargoes. So the cargoes which are seen inside a salivary exosome is shown here. So this is the lipid membrane bound vesicle which carries the lectin, calmodulin, P-CHOP proteins, enzymes, nucleic acids, and all these are present inside the exosome. So exosomes are found in other body fluids as well, such as blood, urine, breast milk and cerebrospinal fluids. Exosomes can modulate intercellular signaling and cellular homeostasis via transporting information-rich molecular cargoes between cells of origin and other tissues. This is how the exosomes act as a signaling mediator. And the lipid bilayer of exosome act as a natural barrier to proteases and ribonucleases that would otherwise degrade their contents. So as I said, it is a lipid enclosed vesicle which contains lot of cargoes. So those cargoes are protected because of the lipid bilayer, which is otherwise degraded by these enzymes. And this makes exosomes an attractive source of material for diagnostic procedures. And what is the mechanistic link between the salivary exosomes and systemic diseases? So cancer-derived exosomes, which are now known to be a rich source of information that reflects the genetic composition and status of their parent tumors. So parent tumors is where the tumor originated, and these cancer-derived exosomes may help in detecting those. Cancer-derived messengers RNAs are the cargo of exosome and likely reach the salivary gland via circulation. So this can reach the salivary gland via the circulation. So I just showed the picture. And there are two types of salivary exosomes released into the saliva that carry cargoes that include cancer-derived products. One is the cancer-derived exosomes, which is released through exocytosis, and the other one is the acinus-derived exosomes, which are released through the fusion of multivesicular bodies in the plasma membrane. So here we can see there is a cancer here in the pancreas. So the cancer-derived exosomes reach the circulation and goes near the salivary gland. Where the exosomes can either come through the acinar cell by fusing with the plasma membrane and coming out as a acinar-derived exosomes, or it can either directly enter through the circulation and then it can come out. So the acinus-derived exosomes and cancer-derived exosomes both reach the salivary gland and they are excreted in the saliva. So this is how the cancer-related salivary exosomes can reach the salivary gland. And talking about the liquid biopsy. Liquid biopsy we can do with the blood, urine, saliva. So all these comes under liquid biopsy, and here we are talking about the salivary liquid biopsy. So it is key to early detection in identification of biomarkers, and this is facilitated by obtaining liquid biopsy samples that contain tumor-derived material such as circulating tumor cell, that is CTC, circulating tumor DNA, that is ctDNA, and circulating extracellular vesicles. These are the things which can be found in the saliva. And the development of multi-cancer early detection tests, which contain probes for drivers of diverse cancer types, will enhance the ability of clinicians to screen individual patients for more than one cancer simultaneously. So all these things, if it is translated, then we can screen the patients for more than one cancer at the same time. And there is a thing called electric field induced release and measurement, EFIRM, which is an electrochemical based platform for rapid non-invasive detection of molecular targets in the body fluids. So here there is a cyclic square wave of electric field. The electric field is in the form of a cyclic square, which is applied to release and detect the mutations. Here what happens, first there is a electrode which has the capture probe. So always during a test or even for ELISA, we need something to capture and something to report. So here also there is a capture probe and there is a detector probe. See, these two are already present there. Which is the capture probe is coated to the electrode, and then when there is a electric field applied, the sample is added and the sample's DNA is linked to the probe, capture probe, and then which is detected by the detector probe. The detector probe is then using the horseradish and streptavidin, then it shows the activity that sample has that particular circulating tumor DNA. So this is how this electric field EFIRM is used. So the hybridized sequences are measured on the electrochemical sensor with a capture probe pre-coated in the conducting polymer. And the HRP labeled reporter probe generates amperometric signals when there is a reaction with the tetramethylbenzene substrate under -200 millivolts electric field. So because of its high sensitivity, EFIRM can detect single nucleotide variants in complex biofluids rapidly. And combined with rapid detection time, that is less than approximately 30 minutes, and a low volume, only 40 microliters of is required, this positions EFIRM at the leading edge in terms of biomarker detection from biofluids. So a study found that the saliva EFIRM liquid biopsy was able to detect an increase in the recurrence related to T790M circulating tumor DNA in 2 of 26 patients. Whereas this signal was missed using the digital droplet polymerase chain reaction and next generation sequencing. So when they screen the patients for any recurrence using both the qPCR as well as the liquid biopsy, here they used the saliva liquid biopsy, which was able to detect the patients who had this recurrence in this circulating tumor DNA in 2 out of 26 patients. And recently we have just seen about the SARS-CoV-2 testing. There are many techniques which were used, and now we are going to see about the saliva-based SARS-CoV-2 testing. So a favorable public view of saliva testing for COVID-19 and both the saliva direct test and the Spectrum Solutions SDNA 1000 test have been submitted to US FDA use authorization. So they have submitted this for saliva-based SARS-CoV-2. And highlighting the utility of saliva testing, the University of California, Los Angeles created and deployed swab sequence kits campus-wide. So they swab sequence kits were also given. And saliva also could be used to provide information regarding the infectivity and the immune response to this pathogen. So not only did they detect the presence of the virus in the saliva, but they can also find the immune response of the host, whether the person has already developed the antibodies for that. So the EFIRM test is capable of detecting either viral antigen and post-neutralizing antibodies as well as viral genomic RNA, provides a 360-degree view of a person's infectious state and immune response. And what are the future perspectives of salivary diagnostics? So the 3 EFIRM based assays have obtained the clinical improvement amendments. So the clinical improvement amendment, they get a certificate for finding a diagnostic solution for a particular disease. And these are the EGFR exon detection, T790M in plasma, as I said, and there was EGFR exon, T790 in saliva, and SARS-CoV-2 immunoglobulin GS1 in saliva. These were the tests which have obtained the certification as of now. And commercial partnerships are required to take the EFIRM test to the next stage. So there should be a synergy between the basic and translation research, which is a key driver of tests moving towards regulatory approval. So saliva screening is much less invasive and does not provoke a strong patient reflex. An increasing number of studies have been showing the sensitivity of saliva at detecting the SARS-CoV-2 compared with the PCR-based nasal swabs. So these are the future directions of salivary diagnostics. So these fundamental biological discoveries have accelerated the application of saliva-omics to the clinical environment, and the remarkable redistribution of exosomes from the diseased tissue to the saliva via the circulatory system can be exploited for diagnostics, patient stratification and the treatment monitoring.